can we measure pain? We all know what it feels like, but pain that seems intense to one person might feel mild to someone else. Pain might be a personal feeling, and there is no magic number or perfect way to describe it.
So how can we investigate it and make it objective?
Enter Flore Van Olmen, the PhD student of today's episode. Hailing from the Voets Lab, Flore is the subject of my lens. Seeing her work, her collaboration within the hospital with clinicians, her research in the lab, will allow me to bust the myth. Pain cannot be measured. I once again stepped in the landscape of the researcher to fully analyze and understand their work from a close distance. How can you measure pain? What would be its application in disease? To perceive pain, our body uses sensory neurons to notice something painful and inform the brain to react to the stimulus. The way neurons communicate is pivotal in pain detection.
Neurons do this by quickly opening up pain channels, little gates that open up and allow calcium into the cells. This causes an electrical signal that rushes to the brain,
and that's when you feel pain.
Pain can be caused by many different factors, such as certain diseases, genetic changes, and inflammation. And these could be recreated in animals and cells, allowing us to uncover the mechanisms behind them and even test possible treatments. Flore tries to investigate these causes by, for example, activating altered neurons and evaluate the amount of calcium that is transmitted.
We could see that the amount of calcium could be a good piece to really quantify pain, but how would this look beyond the lab dish? To establish this connection, Flore shows how both animals and humans can be tested for these pain differences.
In mice, we can mimic certain diseases or genetic changes and investigate pain behavior.
Flore tests how they react to different stimuli, such as a small needle or a hot surface. By having this zoomed out look on mice with the exact same type of neurons as shown before, Flore can link her findings to behavior. But how would we apply this to humans and disease? The search for these answers makes it that no 2 days are the same for Flore. To describe pain in patients, Flore uses different techniques to measure all aspects of pain, such as sensitivity to temperature or even pressure. In a way, pain truly becomes a number we can analyze. Step by step, using a precise and focused approach, her research measures the different aspects of pain, revealing the subtle signals the body sends long before pain is felt. Doing so, pain becomes not just a symptom, but a key, unlocking new ways to heal. This is the wild lab life, and this is just one of the many stories here that are a part of the great age of neuroscience.